And good morning to you all. My presentation is modeling bacteria transport and fate, investigating the impact of soil water repellency dynamics. Um, Bacteria studies are conducted with two aims: controlling bacterial soil and water pollution and bioaugmentation processes for groundwater bioremediation. That's interesting to know that the first aim is almost to retain bacteria in the environment or in soil, but for bioaugmentation is injection of bacteria to get a certain depth of soil to remove contaminant. Bacteria transport mechanisms are transport through different mechanisms. Bacteria are transported through different mechanisms, including advection and dispersion, and straining, which is physical straining, or bacteria can be absorbed or attached to the surfaces or other surfaces like such contaminant in the soil, or bacteria can be blocked in the soil as a result of the aggregation, or can be biologically attached or provide clusters and called ripening. These mechanisms are influenced by different factors in the soils, and also with regards to bacterial properties and solution chemistry. As we can see, there are a number of bacterial properties, including size and shape, surface charge, hydrophobicity, and concentration. Also, pus media or soil properties, including water velocity, particle size, type of minerals, clay content, organic matter, biofilms, and also liquid properties like pH and temperature and ions. For those four mechanisms, we can see that the interactions of these factors are important. For instance, for advection dispersion, almost we have biological and physical properties that are important. But for attachment or absorption, we see that it's quite complex. Biological, chemical, and physical properties, including bacteria, soil, and liquids, are important. While for blocking and ripening, which are almost biological one, bacteria cell properties can be very important. Among all these properties, it seems that our studies show that water content plays an important role in fate of bacteria, and bacteria transport increases with increase of water content, or retention of bacteria is increased with a decrease in water content. Subsequently, another properties, which is soil water repellency, and is the main topic of our presentation today, highly impacts water content of soil. So to know soil water repellency, we need to define wettability. Wettability is the ability of a liquid to spread on a solid surface. So water repellency is not limited to soil, but can be applied for medical, industrial aspects, and also for engineering. So there are many applications for water repellency: optical devices, textiles, airplanes. So our expertise is remediation or control of, let's say, bacteria pollution. But soil water repellency maybe can be defined very well with contact angle. If soil water contact angle is 0, then the soil is wettable. However, when the contact angle is between 0 and 90, soil is called subcritically water repellent, But when contact angle is higher than 90, then the soil is classified as water repellent soil. Wettability of soil is mainly controlled by organic matter, but hydrophobic organic matter. So this means that uh, quality of organic matter is really important for soil wettability. One further important point regarding soil water repellency is that it's a temporary property and it appears when the water content of soil is lower than a certain content, and sometimes it appears at a certain temperature. So although it's a temporal property, we see that this property impacts many phenomena of soil and many the fate of the many reactions and properties in soil. So let's come back to bacterial transport. Traditionally, bacterial transport is done either through saturated flow condition or unsaturated flow conditions. I mean, at the column scale and in separate columns. So when we have saturated columns, we eliminate the air interfaces, and then we investigate the ability and the capability of the solid liquid absorption or carrying contacts or such pore size and bacterial like bacteria blocking impacts. But for unsaturated flow, we have all these interfaces in the soil, and of course, it would be more complex. And I began this field of study many years ago, and also I focused on conventional saturation and on unsaturated and unsaturated columns. For instance, this is the first study I worked and tried to understand how chemical component of soil impact bacterial transport. Or these two studies, I tried to understand how bacteria are released from manures and organic waste, and then they are transported through different soil texture. For instance, here we have picture curves, which is the changes of bacteria concentration versus pore volume. We see here that the cow manure release higher concentration of bacteria compared to poultry and sheep manures. And when manure is applied into the soil, we see that how bacteria concentration is increased. This is the control, for instance, and this is the retention profile of bacteria into the soil for sandy soil. And then we compare different texture, and we see that, for instance, silty clay loam could retain a greater concentration of bacteria through soil comparison to sandy soil, which is logic. But while I was working on this topic, I was thinking that whether we can design an experiment to consider both favorable and unfavorable conditions so that we are able to recognize different mechanisms of bacteria simultaneously or not. So I saw at that time some experiments that provided unrealistic initial boundary conditions to support or to understand the mechanisms of bacteria. For instance, increasing the level of pH so high so that they can eliminate one mechanism. But that was something that I was thinking that how we can provide such experiment to have all these at the same time. We should be able to recognize different mechanisms. So I designed this experiment, and I refer to again to soil water repellency constant. So water repellency is appear when the water content in soil is low. So I thought that when soil columns are dry, and then if we impose one pulse of bacteria or tracers, and then we leach for a certain time, and then again we add another subsequent bacteria or bromide with the same concentration, we are able to recognize and to differentiate the mechanisms for one, actually for the whole system, including pulse 1 and pulse 2 together, so that we see the general behavior of the soil, and also we are able to model or to recognize each pulse separately. So I conducted these studies for sand as a systematic studies, and also for soil, natural soils, including vegetable and also water repellent. So these are the figures from systematic studies, sand environment. So on the left side, we have vegetable sand. On the right side, we have water repellents. The upper figures shows bromide breakthrough curves, and the lower figures shows the bacteria curves, bacteria breakthrough curves. So these symbols show the observed data, and the lines show modeling. Here we have bromide transport through vegetable soil, and we see that the data was nicely captured by model, but at the same time, our experimental data shows that as soon as the effluent appears at the bottom of the columns, we have the highest concentration, and then the concentration sharply decreased and until the end of the first pulse. But when we injected the second pulse, the concentration increased, and we got the second peak, and but again decreased until the end of the experiment. So the second peak was actually, it seems that it's diluted, very likely due to the increase of water to the system. This was almost so and true for water repellent soil, but the falling slope was almost a little bit, say more slowly than the water repellent soil, vegetable soil. But one interesting point was that after changes of water repellency situation or states in the soil, we saw that the model was not able to predict or to capture the falling slope for bromine. So we used these data. I mean, we optimized hydraulic properties using bromide transport, and then included them for bacterial transport. Here we have bacterial transport through vegetable sand, and here we have bacterial transport through water repellent sand columns. So E. coli data was nicely captured for both pulses, and very similar behavior to bromide it had, but Rhodococcus erythropolis, that was hydrophobic bacteria, in comparison to E. coli that was hydrophilic. Actually, we see here that it shows scatter patterns. This was increased, I mean, scatter patterns in tendency for water repellent soil, but we were able to model Rhodococcus transport. But again, as we see, the data are very scattered. However, we were not able to model E. coli transport. We see that the retention has been very high. So that was a big problem for us, and we were not able to model both pulses together when soil was water repellent. So we then went to another scenario, try to model each pulse separately. So please note that the model actually was run once more. So we simulated the data according to the further scenarios, meaning that, for instance, the initial water content of these pulse was dry, but this part was not very close to saturation. So for wettable soil, which is on the left side, the data was captured by model very nicely. But also this is for bromide, but also on the right side for wettable. For the first pulse, we could actually simulate the data very well. However, when we have disturbances and we have changes in water repellency for the second pulse, this is also for, as I said, for bromide, but the peak rising and falling slope was not predicted very well. With this, we predicted bacteria for each pulses. Here we have the data. So E. coli and Rhodococcus for wettable soil on the left side was nicely captured by model, but we see here Rhodococcus. The data actually was very scattered, but at the same time we were able to model. So E. coli was possible to be modeled here for each pulse was interesting, while we were not able to model it for when we have two pulses together. Rhodococcus second pulse was captured again, and also we were able to model the second pulse. This data actually can be summarized in three tables. Here we have simultaneous simulation of pulses 1 and 2, when we have two pulses together. And on the right side, we have pulse simulation for each pulse. So for the first scenario that we simulated two pulses together, here we have wettable sand and water repellent sand. And we found that straining, which was physical retention mechanism, was higher than attachment, both for E. coli and Rhodococcus in wettable sand. But also this was for E. coli in water repellent sand, but not that much for Rhodococcus in water repellent sand. It is interesting to say that Rhodococcus was physically retained in wettable sand. However, attachment coefficient, that was physicochemical mechanism, was really low. The values were very small. But we see that detachment, that is also physicochemical mechanism, was really high. That was a question. That's why when we have such low rate of the attachment, why should we have such high rate of detachment? So that was a very logic question to run and those experiments, and also to have separate simulation. Here we have the separate simulations. Table A on the right side shows for wettable sand, and table B shows for water repellent sand. So attachment and detachment as a physical chemical properties, and straining and release as a physical properties, physical mechanisms. So coefficient for attachment for E. coli was higher than straining as physical. So this means that when the soil or the sand columns were dry, E. coli was attached to the particles. But interestingly, Rhodococcus physically strained to the particles or in the columns. But when the soil actually was wetted, or the water content increased into the columns, we see that these the mechanisms flip between attachment and detachment for bacteria, so that E. coli was strained, but Rhodococcus attached to the particles. So as we see here, that detachment for Rhodococcus was really high, and that was reflected in our simulation. We saw that how the data was scattered. So this data shows that mechanisms are different for different bacteria with different hydrophobicity and hydrophilicity, while system was dry and wet with different degree of saturation simultaneously. But when we look at water repellency, it was interesting to see that except one case, that was for E. coli with high retention, all data shows that bacteria actually was strained. So straining as a physical mechanism has been the dominant mechanism. For instance, 1-2 point for E. coli, and then point in comparison to 0.3 for attachment, and 0.27 compared to 10 to minus 5, and then 0.4 comparison to 0.6, that was an exceptional that I will explain, or 24 comparison to 5.45. So that was a clear answer for us that when we have a water repellent soil, very likely bacteria are strained rather than going to any attachment. But when we look at E. coli and Androdococcus for the second pulse, we see that the coefficients uh, we don't see that much differences between the coefficients, particularly for E. coli, and we saw how highly it was retained. So that was something that we look at release factors, and we see here that the release was higher, physical release was higher than detachment. So we concluded that very likely E. coli has been also retained physically. Yes, this was for systematic studies. I forgot to mention that for E. coli, that we were not able to model the data for breakthrough curves, we use profile retention data using, because more than 70% of the bacteria was retained in the soil profile. So we use profile when we were not able to simulate breakthrough curves. We also calculated further properties as the auxiliary parameters, such as we calculated hydrophobic interaction energies for both bacteria to realize how bacteria are interacting with air-water interfaces. We found very negative values of hydrophobic interaction potential for Rhodococcus comparison to E. coli, and for distances less than, say, 2 nanometers, if I am right. But this means that very negative values of the interactions shows that very likely Rhodococcus can interact with air interfaces very much higher than E. coli, and E. coli needs to be very close to the interfaces. So that was very likely a reason that we saw such scatter patterns in transport of Rhodococcus, so that these interfaces, I mean air-water interfaces, as the can be changes through the infiltration process, and those bacteria can be released from those interfaces. We also calculated capillary potential energy profiles for each bacteria, so that we found that at distances less than 395 or 400, I would say, Rhodococcus can be trapped to the water films, but later on, at larger distances, say larger than 400, E. coli was retained, and very likely Rhodococcus was released. So with this, for our systematic studies, we found that attachment of bacteria, let's say, was really low in wettable soil, but straining was really high for water repellent soil sands. But when we model each pulse, we found that these mechanisms are different for each bacterium. E. coli attach, but Rhodococcus strain in dry wettable sand, but E. coli, when this wettable sand became wet or became more saturated, E. coli strain and Rhodococcus attach. So meaning that meaning that E. coli was physicochemically retained in the dry condition of sand columns, but Rhodococcus strain, while when the soil got saturated, E. coli strain and Rhodococcus attached. But when the sand actually was water repellent, straining was the dominant mechanisms for bacteria. And also through those interaction energies and capillary energies, we found that Rhodococcus was trapped in and released from water prints sooner than E. coli, and that was due to the hydrophobic and also size of bacteria. The result of these data are published in 2 papers, and the links is also provided here, and you can get more information about these studies in more detail. So another aspect of this idea was to also generalize and extend the idea to natural soil. Was it true that we can get the same results and the same trend for bacterial behavior and bacterial transport when we use natural soil, I mean wettable and water-bearing soil, or not? So we conducted actually a similar setup when the columns were dry, and we injected bacteria, imposed the first pulse. Again, we injected the second pulse and continued the leaching. I should add that I have been working on 2 flow conditions: saturated flow, meaning that I imposed saturated flow using tension disk infiltrometer. So the water was flowing into the columns under saturated condition, but the columns were not saturated or were dry, I would say. But also we imposed unsaturated flow, -5 centimeter, and then the infiltration was conducting under dry condition of soil for these conditions. So I summarize or say focus on saturated flow in this talk, preventing or avoiding any confusion. So we continue this, meaning that I will focus on saturated flow. So the idea was that whether we saw the same trend and we see that whether we have changes in retention and transport of bacteria and the mechanism and what's the main mechanisms of bacterial retention when we have wettable and water repellent soils with regards to such initially boundary conditions and water repellency. So here we have Brutu curves of bromide, and for wettable soil on left side, and on the other side, the right side, we have the data for water repellent soil. So the pulse, the symbols showing the experimental data, and the lines are showing model. So we see that we were able to model the data for the first pulse for the wettable soil for bromide, but not nicely for the second pulse around the peak time. So we see that the model predicted the peak sooner than expected. But interestingly, we were able to model the second pulse very much better than the first pulse. This means that when we have water repellency into soil, that would be a problem for prediction of these prediction of the bromide transport. So then we conducted modeling for the separate scenarios. I mean, for the simulation of either pulse separately. So For wettable soil, nicely the data was captured, but for rising fall, for the second pulse, we were able to model that, but not for the falling slope, and we have our overestimation here for bromide transport. But interestingly, when we separate each pulses for water repellent soil, the data was captured nicely, I believe, although we had a little bit overestimation at the end of the experiment. These are signs of water repellency cessations, I believe. So when we have changes in water repellency, such I would say trend can be possible, and these are coming and originated from the water repellency cessation and breakdown. So we also modeled a bacteria for each soil, and here on the left side, we have transport of bacteria, which is modeled for wettable soil, and on the right side, we have transport of bacteria through water repellent soil. Green symbol shows E. coli, and the red symbol shows erythropolis, Rhodococcus erythropolis. So very nicely, we were able to model bacteria transport from natural soil, wettable soil, while we have very high retention. So we see that the retention has been really high. But interestingly, we were also able to model E. coli transport through water repellent soil for both pulses. And although we have such changes in concentration. However, as I said, for bromine, we see that the transport of bacteria, for particularly for rhodococcus, is really scattered, and it was hard to simulate the first pulse when we have water repellence in soil, very likely. But afterward, when water repellency very likely stopped, we have nicely modeled these data. For each pulse, we were not able to model bacterial transfer for water repellent soil, although here we could model the two pulses together. So here we have modeling for wettable soil only, and for E. coli, the upper figures and the lower figures showing rhodococcus. So the modeling was okay, but I believe it was only 50%, but our statistical analysis shows that it is promising and it is good enough and can be trusted in terms of the data and validation. So we could also get these peak, although the retention was high for pulses, but not very good for rhodococcus, as I actually mentioned earlier. We have always such scatter patterns in rhodococcus transport. So all in all, if I want to summarize the mechanisms of bacterial transport for wettable soil and water-repellent soil, we found that attachment was really higher than the straining in natural soil, and that was very interesting in comparison to the sand experiment. So when we had wettable soil and water-repellent soil, we had attachment as the main mechanisms of bacterial retention. But at the same time, wettable soil could retain bacteria much more than a water-repellent soil. So E. coli was retained higher than rhodococcus when we had attachment, and when we had straining, there was no significant difference between the mechanisms and between retention of E. coli and rhodococcus for wettable soil. And this was so for straining in water repellent soil. But We also look at detachment and release. So detachment refers to physical chemical detachment, and release refers to physical release, meaning that bacteria are very likely are actually coming from pores that are blocked, and then suddenly the pores are opened, and I mean when bacteria are strained there, and so we compare detachment for wettable soil and water repellent soil. We interestingly found that detachment for water repellent soil was really higher than the wettable soil, and that is something interesting, and comparison to physical release. So with this, we can also compare natural soil comparison to sand, and in comparison to the to compare mass recovery. So we compare and we analyze the mass that can be recovered in the effluent or retention profiles. So here we have natural soil. We see that in the first pulse, when the columns were dry, only 3% of E. coli was actually transported under saturated condition, and when the soil was wettable. But that was very interesting that only less than 1% of bacteria or E. coli was transported in water-bearing soil. So when soils are dry, so they can retain a great portion of bacteria that are injected. And Rhodococcus was actually transported higher than E. coli, was interesting, and 20% when the soil was dry, but for water repellent soil, in comparison wettable soil, it was for the 74%. That was really high. So if we compare these data to sand studies, we see a big difference. For instance, here we have 70% of E. coli that are transported, but here only 3%. And then here 30%, it's 129. So when we are working in bacterial transport studies, we should be careful that to consider this, that we shouldn't rely only on systematic studies, and we should also consider initial conditions of soil and soil properties, and we cannot generalize the data for uh, coming from sand data. So if we look at water repellent soil as well, here only 6% in the dry pulse was transported. Here for E. coli, 1%. So this means that natural soils have high capacity, although they were or they are water repellent, they could retain a great portion of bacteria. But whether they could retain those when they become saturated or very close to saturation point or not. So we see not. About over 100% of bacteria, very likely a portion of them are coming from the first pulse. Over 100% of bacteria are released. So this slide was to compare systematic studies and natural soil in terms of the mass recovery. The result of these two, let's say, studies for experimental data and also modeling of these data are published in Science of the Total Environment, and the latter 2024 was published a couple of days ago. So all in all, in bacterial transport studies, it is crucial to evaluate different important properties, such as running experiments, correct experiment with regards to our aims, running modeling, and also considering mass balance information, interaction energies, and we should consider bacteria properties, size, shape, and macromolecules. And also we should benefit from chemical tracers, which is also traditional in transport studies. All in all, our data shows that bacteria attach to natural soils rather than undergoing significant straining, and that was very different from systematic studies. And attachment was called in inevitable soil very much higher than water repellent soil. And our data, particularly interaction energies and modeling, show that size of bacteria is important, and also hydrophobicity of bacteria are important, so that they impact the dominant retention or release mechanism. So that a small hydrophobic Rhodococcus erythropoiesis detached means that it was interacting physicochemically, while large hydrophilic E. coli was released. So at the same time, through our modeling, we found that water repellency caused selective bacterial physical release or physical detachment, as I mentioned now. And our strategy and scenarios using separate optimization of bacterial pulses highlighted physical straining and release rules. So that was something that we were able to follow and could approve that how straining and release can be modeled. I have been working with different universities, and my projects have been funded through different agencies and funders, including when I was in Germany during DAD, Humboldt Fellowship, and also when I came to UK through Marie Curie Fellowship. Also, I work with Leibniz University of Hanover, and also different universities that are here. I also would like to thank to ISMC and the research committee for the ISMC 2024 Young Scientist Award that was donated to me. I appreciate their consideration, and I'm honored to having this award. At the end, I would like to thank you all and all my colleagues during all these years that helped me and cooperate with me for this project. Thank you.